Chapter 8, Molecular Aspects of Microbial Growth. Now, up till now, we've discussed microbial growth in terms of, in loose terms of metabolism and things like that. Here we're going to be looking at specific metabolic functions that have to occur for the cells to go from 1 to 2, what genetic structures have to be there, what genes have to be expressed, and all of that. We're going to be looking at more than just the binary fission, 1 to 2. We're going to be looking at, as you see here, things like, here, A, formation of endospores. Here, the orange is the cell, the green is the endospore being formed. It's being stained here in this fluorescence micrograph picture. Or, in B here, they went and they stained genetic regions. The blue and the green are the ORIC, ORIC, which is the origin of replication for the chromosome region. As the chromosome replicates, there's the replication initiation part, there's the other. So you end up with 2 circular chromosomes being pulled apart. Or as they're showing here, as the genomes have been pulled apart, you're going to start to see new cell walls, septums, being made to separate the cells. And then how do the cells elongate, get bigger? Here you're looking at Staphylococcus aureus. As the round cocci elongates a bit and then splits down the middle. What are the proteins involved there? What's the regulation of that? That's what we're going to be talking about. Now keep in mind, a lot of these pictures you're going to be looking at either the microscope pictures or the artist rendition of microscope pictures are going to be based on what is known as super resolution microscopy. Okay, this is the best of the best microscopes with the best lenses possible, backed up with the best computer systems possible. Resolution is down into the 5 to 50 nanometers. I'll put things in perspective. About the best our light microscopes upstairs can do. I mean, you can barely see the 1 micron gram-positive cells. I mean, stain things right, you might be able to see less than 1 micron. So this is 20 times better magnification, 100 times better magnification, and still be considered a light microscope. What they've done is they've used, for a lot of these, they're using reporter genes. The gene of interest that has a little something added into the gene at the end. A fluorescent, a part of a protein that will not interfere with the overall folding of the gene product, so you get a working enzyme or a working structural protein, but at one end of it, the N terminal or the C terminal, you are going to see, part of what's called green fluorescent protein, GFP. GFP under certain wavelengths glows green. This super resolution. They can also tag certain proteins with molecules that glow colored. You flip a switch and instead of white light, like the LED light shining in my face, you get light that's in the UV spectrum. Hits one of these fluorescent markers, and that fluorescent marker will then glow green. Flip another switch and that light, instead of being the UV, it's now in the infrared, somewhere in the red spectrum. A different marker, different fluorescent marker, is now going to glow, be excited and glow yellow. Flip the switch again and you now have yellow light, a wavelength that's, and now you're getting something that's, a different marker that's going to fluoresce something, whatever. And that's how these different photoactivated probes work. multiple bulbs, that can, emit at specific wavelengths, or one white light one that has, the ability to, the microscope has the ability to send it through different filters so that only certain wavelengths of that light will pass through. Kind of similar to what you've done with the Spec 20, but on a much finer scale. So that way you can take multiple pictures of the same thing, as you see here. These are single cells. And notice the reds, the greens, the blues. I think there's even some yellows down there. All you have to do is get this into focus and then hit a button. Take a picture of everything under white light. Hit another button. Now it's just certain wavelength. Red. Hit the button, another button. Different wavelength coming out from the light. You see the blues. Hit another button. Different wavelength. Now comes the greens, and so on. So this will allow, to look at one singular picture or one singular view in a microscope and be able to discern the location of numerous proteins or numerous gene sequences in a chromosome that's either in a nucleus or here it's in the nucleoid.